Well, hello everyone, and welcome back to Human Biology here at Chaminade University. Today we will be discussing cellular metabolism. So just a brief overview of cellular respiration. Basically, all of the energy that we use for life is obtained by breaking down molecules that were originally produced in plants. So what molecules is it that we eat, right? So we're going to be eating things like sugars, fats, nucleic acids, and proteins. And all of them were created by using the energy that's stored in ATP. So the energy in ATP can be formed by breaking down carbon to hydrogen bonds. So every time we break a bond, we store the energy released by that broken bond in the third phosphate of ATP. So ADP would be diphosphate, it would have 2. ATP would have 3 phosphates, and that's the higher energy version. So lower energy di, higher energy triphosphate. So every time we break a carbon-hydrogen bond, we store that energy in the third phosphate bond here of ATP, and then we use that later on to drive metabolic reactions that need energy to be put in, right? And so all the energy that we get comes from breaking down the food that we eat. So we process food and we break it down and we turn it into energy that we can use. And we do that through the process of cellular respiration. Now, ATP, which I just spoke about previously as the primary energy molecule, is known as adenosine triphosphates. So here we have the nucleotide, it's the adenine. This is the same A from A, T, C, and G found in DNA, just FYI. And when we put it with the sugar ribose, so here we have ribose and a nucleotide base, and then 3 phosphates. That makes it a triphosphate. If we only saw 2 here, if it stopped right there, it would make it a diphosphate. Triphosphate is the high energy form, diphosphate is the low energy form, and it readily goes back and forth between ADP and ATP. Now, when energy is released from bonds that are in the surrounding area, it is captured in the form of ATP. So we start off with ADP and free phosphate, and then the energy is utilized to drive that reaction once this reaction occurs. So energy can be released from any molecule that has a carbon-hydrogen bond, such as something like methane, right? Or glucose, or fatty acids. So anything that you eat that has carbon in it, bound to hydrogen somewhere, we can utilize the breakage of that bond to drive a cellular reaction, and we will store that energy in the form of ATP until we need it. Okay, during the process of cellular respiration, we have the gain of electrons for one molecule and the loss of electrons for another. And the way to remember which one is which is this little phrase down here. Leo the lion says grr. So oxidation is when we lose electrons. Loss of electrons is oxidation. Gain of electrons is reduction. And these usually happen so quickly and at the same time, right? One of them is going to be oxidized, the other one's going to be reduced, that we call them redox reactions. A reaction is basically, so redox. One of them's getting reduced, the other one's getting oxidized at the same thing. And this happens quite often during cellular respiration where electrons or hydrogen ions are moved or bounced back and forth, from one molecule to another. Now, not all food that you eat is created equal. Some food gives you more energy than others, right? And giving energy and giving nutrients aren't necessarily the same thing. As you'll see right here, that a gram of alcohol has 7 calories, however, has 0 nutrient value. Whereas a gram of carbohydrates is only going to give you 4 calories. Protein, same thing, 4 calories. And a gram of fat, that's going to be our highest bang for our buck at 9 calories. So if we were to eat a pound of fat, that would give us about 35,000 calories. That would be a whole block of butter, essentially. So a block of butter would be about 2 of your daily servings of calories, not at all the daily servings of fat, though, obviously. Okay, so basically my point is that you are going to be taking in food from the external environment, breaking it down and utilizing it to drive energy. And we do this inside our body by the process called metabolism. And metabolism is broken down into multiple different components. One set is called aerobic respiration. So we have aerobic respiration and anaerobic respiration. If we are utilizing oxygen, it's aerobic. If we are not utilizing oxygen, it's anaerobic. And evolutionarily speaking, the usage of oxygen gave organisms a great leap and bound over the other organisms around them that were not utilizing oxygen. Why? Because the same glucose molecule under anaerobic respiration will produce about 4 ATP. but under aerobic respiration will produce over 30. So it's almost a tenfold, it's 9 and change fold increase in the amount of energy that you would get out of the exact same glucose molecule. So you can see that being that much more energy efficient would give that organism a great advantage over the organisms around them. So aerobic respiration gave organisms a great hereditary or evolutionary advantage. All right, so aerobic respiration requires oxygen and glucose. So we start off with glucose. You may recognize glucose here as C6H12O6. Just as a side note, anytime you see that the hydrogen is double the amount of carbon and oxygen, so you have 3 molecules or 3 atoms, carbon, hydrogen, oxygen, at a 1 to 2 to 1 ratio, you're always dealing with a sugar. Glucose is no different here. It's a sugar, 6 to 12 to 6. Again, C6H12O6. We're going to combine that with oxygen here, and then we're going to convert that over into carbon dioxide and water plus free ATP. So we're taking this complex structure, and we're breaking this really complex structure down into 6 very simple structures and releasing energy. When we release that energy, we have ADP free here, which isn't really shown, and free phosphate that are binding to make this ATP. So again, the energy is stored in that third phosphate bond between the second and third phosphates. All right, and then we're going to utilize that energy elsewhere from another metabolic processes, right? All right, so cellular respiration has 2 major stages. We have glycolysis and the Krebs cycle. Now, prior to having aerobic respiration, glycolysis was all we got. It does not require oxygen, so it's anaerobic. It occurs in the cytoplasm. It does not require mitochondria. Now, the Krebs cycle requires oxygen and occurs in the mitochondria. So we have to have mitochondria present, and this is why certain cells in our body have more mitochondria than others. If that cell has higher energy needs, it's going to have more mitochondria than a cell that has lower energy needs. All right, so within the mitochondria, we're going to break down the byproducts of glycolysis. So cellular respiration starts with glucose, C6H12O6, and it's going to break it into 2,3. So we start with a 6 carbon sugar. We're going to break it down into 2 3 carbon molecules. Those 3 carbon molecules are going to go into the Krebs cycle and get broken down. That's the end product of glycolysis, goes into the Krebs cycle and get broken down into electrons and protons, etc., until it's all the way broken down into carbon dioxide and free water. And we use this to create ATP. All right, so how do we do this? This is what's called a redox reaction or an oxidation reduction reaction. We basically are going to have the transfer of electrons from one molecule to the other. So this one is the reducing agent, which is then transferring molecules over here to the reduced molecule. If we were to be going the opposite direction, right, we'd be going from an oxidizing agent to an oxidized molecule. So basically, losing an electron is oxidation, and gaining an electron is reduction. But again, they happen together, so we call them a redox reaction because at the same time that one molecule is gaining electrons, the other one is losing them. All right, so now we are looking inside the mitochondria into the Krebs cycle. The very first stages of cellular respiration happen outside the mitochondria here. Again, that's glycolysis. That's where we're taking glucose, which is 6 carbons, and turning it into 2 pyruvates, which are 3 carbons. These 2 3-carbon molecules are going to be oxidized inside the center of the mitochondria into something called acetyl-CoA and shoved into the Krebs cycle. As they go through the multiple stages of the Krebs cycle, they're going to be creating NADH and FADH2, which I haven't talked about yet. So let's talk about that for a second. We talked about ATP being the energy carrier molecule for the cell. So if you picture this as like money, right, currency, ATP might be your $20 bill, our most commonly used one. But FADH and NADH, these are 5s and singles, or coins, if you will, right? And so during the cycle, we're going to be making a bunch of loose change in smaller bills that then are going to get converted out through the electron transport chain. They go to the coin star, right, and turn it into bigger money. So the NADH and FADH2 are going to add up to some of the ATP that are going to be released here. Okay, but overall, as we go through the Krebs cycle, we're going to create another 30 or so ATP for each molecule of glucose. So cellular respiration begins with glycolysis. It's our very first step. Glycolysis occurs in the cytoplasm, and what's the process of breaking down glucose, which is a 6-carbon sugar, into a 3-carbon molecule. If you do not have mitochondria, it's going to be the main energy source. It's going to be the best that you got. You go from 6 to 3, go to the next one, 6 to 3, right? You're going to be getting about 4 ATP, I think, out of this. It's really not very much. But if it's all you've got, you do it over and over, you can get enough ATP. So glycolysis is going to be a series of chemical reactions that breaks one molecule that starts off as a glucose, 6 carbons, into 2 pyruvate molecules that end up as 3 carbons. And every time that we lose complexity from that 6 down to the 2 pyruvate molecules that are 3, right? So we start with 1, 6, we end up with 2, 3. Every time we lose complexity, we're going to release ATP. And then we're going to release energy that's going to then be used to create ATP, right? And that is going to create ATP by the process of phosphorylation, or the addition of phosphate to ADP. So this is our low energy molecule. It's just hanging out in solution, waiting for energy. When energy comes and there's a free phosphate near it, it binds via phosphorylation to make a higher energy molecule, ATP, that we store and can use elsewhere in the cell to drive other metabolic processes. But again, it's not our only form of money. We also can take NAD and make it NADH, which is a higher energy. So lower energy NAD plus, higher energy NADH. And so when electrons and hydrogens are donated over to NADH, NAD making NADH, that's going to be another form of energy that we can utilize in the cell as well. Again, you can consider this your $5 bill instead of your $20. As I mentioned previously, glycolysis is that first step. It's anaerobic, and it's all that anaerobic organisms have, by the way, or organisms that don't have mitochondria as well, I might add. And it only yields a very small amount of ATP. Only about 4 ATP are made for each molecule of glucose that are broken down into pyruvate. But remember, 2 ATP molecules are required to start the process, so that's a net gain of only 2. However, organisms that don't have the ability to utilize oxygen, it's the only way that they can derive their energy from food. Because every organism is capable of carrying out glycolysis. Evolutionarily speaking, it's a lot less complex. Probably the most early ones to evolve. All right, so this is the process of glycolysis. Here we have our 6-carbon sugar here. It's glucose. We're going to draft it as 1, 2, 3, 4, 5, 6. We're just going to keep the carbons here, right? We're going to add 2 ATP in there, which is going to then cause 2 ADP and 2 free phosphate, right? That's going to drive it's going to give the energy needed to drive the reaction, where we're going to flip things around to make fructose bisphosphates. From here, we're going to split that into a phosphoglyceraldehyde, which then gets split in half to make pyruvate. And we're going to, at that point, also release 1 NADH here and 2 ATP. So we put 2 in, we get 2 out, and then we get 2 NADH as well. So the net gain is going to be 2 ATP molecules and 2 NADH molecules. It's really not going to be very energy efficient. But if it's all you've got, then it's better than nothing, right? However, those of us that are able to utilize oxygen are able to then continue on and take pyruvate and further break pyruvate, which is a 3-carbon molecule, down into carbon dioxide, which is a 1-carbon molecule, right? So we're going to take a pyruvate molecule, which is 3 carbons, and make 3 1-carbon molecules. Overall, remember, we're taking a 6-carbon molecule, we're making 2 pyruvate molecules, and eventually making 6 carbon dioxide. The point being that pyruvate, that 3-carbon molecule, still has a lot of energy stored at the end of glycolysis that we can utilize if we send it through the mitochondria. So the first thing we have to do is make acetyl-CoA. And I'm not going to talk you through all of that, but basically we're going to utilize these multiple different stages to create acetyl enzyme CoA. Once acetyl-CoA is created, acetyl-CoA is going to, again, it's going to be the oxidation of pyruvate via the we're going to create 1 NADH here as well, and 1 carbon dioxide is going to come off. So we started off with a 3-carbon molecule. Now we end up with a 2-carbon molecule and 1 free carbon dioxide. Now this acetyl-CoA is going to go into the Krebs cycle. It's going to be the beginning stages of the Krebs cycle. And it's called a cycle because what comes out is actually also going to start back in as well. So acetyl-CoA is going to be sitting around waiting for ATP to become available. When we don't have enough ATP, acetyl-CoA is going to head to the Krebs cycle. If we have enough energy already and we don't need ATP, acetyl-CoA is going to be diverted to fat synthesis and energy storage, which is why if you eat more calories than you burn off, the extra calories are going to be stored as fats because acetyl coenzyme A gets diverted into the adipose tissue. All right, so this scenario is rather long. I will let you read through it, and then I'll let you read through the one at the end on your own as well. I'll give you about a minute to do so. So the overall question here is regarding Fred and his ability to lose weight and whether or not he is able to lose weight if he simply eliminates soda from his diet.
All right, so let's go back to the Krebs cycle. So acetyl coenzyme A is the beginning of the Krebs cycle. It's going to enter the cycle and bind to a 4-carbon molecule, forming another 6-carbon molecule. So remember, we started with 2 because the 2 came from, remember, pyruvate was 3. We lost 1 to carbon dioxide. Now we had 2 left. Those 2 are going to bind to 4. That makes a 6-carbon molecule. You're starting to see something, right? We started with a 6-carbon molecule as well. 2 carbons are removed because carbon dioxide, and electrons are donated to NAD. And then ATP is produced, and then that 4-carbon molecule is recycled. So as I mentioned, it's called a Krebs cycle because the end product goes back in. So the 4-carbon molecule that ends up coming out is what goes in and attaches to acetyl coenzyme A to make the 6-carbon molecule. So this 4, it's just going to be constantly recycled. And every time we're going to be making more and more NADH and FADH2. Remember, these are, you can use a couple analogies, but let's use the coin analogy for now because we're going to go through the coin star when we go through the electron transport chain and come back out with dollar bills. Okay, so here's our Krebs cycle. Here's coenzyme A. It comes in as 2. It combines with a 4-carbon molecule that's known as oxaloacetates. It becomes this 6-carbon molecule. That 6-carbon molecule has a really weird and kind of awkward structure. So the first thing that happens, boom, we're going to cleave that off, making this 5-carbon molecule. Boom, we cleave one more off, making the 4-carbon molecule. That 4-carbon molecule gets recycled again. Yeah, there's a lot of steps inside there, and there's a lot of things coming off here as well. NAD is going to become NADH. That's 1. Let's call it a quarter. Here's another NAD, and here's another quarter, right? So each one of these change, say that's a nickel. Each one of these are going to be changed that were then later on during the electron transport chain going to convert back into ATP, which are our dollar bills. Or I guess if you want to use the 20 and the 5 scenario, these are 5s and 1s that are going to eventually become $20 bills again when we cash them out into the Krebs, into the electron transport chain. Right, so that's kind of the overview of the Krebs cycle. I'm not going to ask you any specific details on what the names of all the different chemical compounds are at every step, but I do want you to know that we are recycling the 4-carbon remains from the last cycle, adding it to the 2-carbon molecule that's coming in terms of acetyl coenzyme A, which forms this weird structure that gets cleaved off very easily. Another one gets cleaved off, and then we get back down to the 4. Each time we're going to result in the creation of 2 carbon dioxide molecules, 2 NAD, 3 NADH, and 1 FAD. Okay, but then what happens to the extra electrons, right? You go through the electron transport chain and what's called chemiosmosis. So mitochondria uses chemiosmosis process to create ATP, and they do that by using proton pumps, which are going to use electrons that have been excited. So they're energetic electrons. These all come from your food molecules, and when they're excited, they get pumped across the cristae. I didn't actually use the word cristae before, so let me go backwards a second. These here are folds in the mitochondrial membrane. What's interesting about mitochondria have 2 membrane. They also have their own DNA. Their DNA is separate from the DNA of the cell. Most of the DNA in the cell is found in the nucleus. Mitochondria have their own DNA that's completely separate. And in terms of human lineages, it only gets passed down through the mother because mitochondria are lost in the sperm tail. But I digress. The point being that this outer membrane here, this light orange or darker orange membrane, that is going to match whatever organism it's in. If it's inside a dog, it's going to match a dog membrane characteristics or a human membrane characteristics, whatever animal it's in. But the internal area is going to have mitochondrial membrane characteristics. Inside the mitochondria maintain their own personal house, basically, their own DNA, their own workings, their own everything. The cell just houses them separately inside the cell and keeps them safe. Point being, these folds here are called cristae. And as we pump the electrons across the cristae, we can start turning that NADH into ATP. Now I'll go back forward again. Sorry about that. I just didn't want to introduce the word cristae without you having heard the word cristae before. All right, so I could have just gone forward, I guess. Here's the electron transport chain and chemiosmosis. Again, this is happening here in the cristae. Basically what's happening is the electrons are jumping off of the NADH. Remember they popped on in the form of electrons to make NADH. Now it's going back to NAD and releasing that electron. Same thing with FAD, releasing the electron and becoming FADH is releasing the electron and becoming FAD. And when those electrons are released, they're going to go through multiple different protein complexes until they finally pop back out in the form of ATP. What do I mean by that? You don't need to know every step in this pathway, but what I want you to understand is that after the Krebs cycle, we get ATP, right? Let's go back to the 20. We'll call this a 20, we'll call this a 5, and we'll call that a single, right? So we have the $20 bill, but we're also going to get 6 5s, 2 singles, and they're going to have to get converted back into 20s, right? So how do we do that? In this case, it's actually electrons that we're converting. The electrons here get caught on the NAD as the form of hydrogen. They get caught on the FAD as the form of hydrogen. So when we go up here, FADH2 is going to release the hydrogen, NADH is going to release the hydrogen. Those are going to get caught through these multiple different complexes where they get popped up and out, and eventually end up forced back through this process here, which contains an enzyme called ATP synthase. This is an ATP synthesis. And we call this, I've been calling this kind of like the coin star, right? It's where you take your 5s and make your 20s, or you take your coins and you make your bills, right? So we take our electrons and we convert them into ATP. Every single time a hydrogen comes through this hydrogen pump, we have ADP and free phosphate sitting ready to go, pop, making ATP. We have 32 free phosphate and 32 ADP, which is the low energy version, and we have 32 hydrogen that come across. And when the hydrogen come across, they pick the DP together with the phosphate to make the ATP. And that's called oxidative phosphorylation. That's the last step in the electron transport chain. And that is how we take FAD and NAD, or FADH and NADH rather, and turn them into ATP. So we're able to get back to the energy currency that everyone else is using elsewhere in the cell. Okay, so how do we make ATP? Well, we need a lot of different biomolecules. We need the precursors for NADH and FADH. If we don't have enough of the NADH, we will never make it over here to the electron transport chain. We'll never be able to drive this proton pump. We'll never be able to make the ATP. So we need NAD+, and we need FAD+. These are vitamins B3 and B2, known as niacin and riboflavin. We also need coenzyme A, right? We saw that earlier. That's called pantothenic acid. That's vitamin B5. The electron transport chain protein has a specialty enzyme called coenzyme Q that's very important. We need omega-3 fatty acids to help keep the electron transport chain functioning. We also need an enzyme called thymine. Thymine is vitamin B1, and it's a coenzyme that's involved in carbon dioxide production during the oxidation of pyruvates. Magnesium is required for ATP function. And this is a list of the different foods that you can eat in order to get all of these different vitamins that you need to make sure that your ATP synthesis is functioning properly. That's going to include fish, chicken, pork, liver, beef. These are a lot of different meats, right? Proteins. Also, veggies like mushrooms and spinach and peas, peanuts, nuts, seeds, avocados, cheeses, eggs, sweet potatoes, etc. All of these are going to be really high in these vitamins that you need to make ATP. What are the other nutrients that produce ATP? Basically, all macromolecules can be broken down and shared and turned into one another, and also turned into ATP, right? There's a lot of different pathways that you can funnel things into to turn them into whatever you're lacking. So if we have polysaccharides, which is the very long version, the polymeric version, we can break them down into the monomeric version of glucose. Or protein, which is again the polymeric version, and amino acids are the monomer. Triglycerides, polymer, fatty acids, and glycerol are the monomers. Nucleic acids are going to be the polymer, nucleotides, monomer. So basically, any one of these macromolecules can be broken down into the biomolecules that we need, the monomers that we need to drive ATP synthesis. This is what I meant when I said that a lot of these different biomolecules have crosstalk, and you can, if you're missing in one thing, you can utilize other pathways to get that product, because we want to make sure that nutritionally we're getting everything we need no matter what we're eating. So nucleic acids go back and forth to nucleotides. Again, all of these guys at the top are going to be the polymers. These guys are going to be the monomers. They're calling them macromolecules and building blocks, but still polymers and monomers. The monomers are nucleotides, amino acids, monosaccharides, glycerols. But we can stick the glycerol over during the process of glycolysis and turn it into pyruvate and feed that in, right? We can take a nucleotide and feed it right on in here through aerobic respiration into the Krebs cycle. We can have deamination, meaning taking the amino group off, deamination, de-aminoing, right? Taking the amino group off of the amino acid and then tossing that in to become pyruvate, or if we break one off of pyruvate, we get acetyl-CoA, so we can hop in at either one of those. So my point is that we don't necessarily need to have glucose to make ATP to feed into this cycle. This is just the fastest way. Right? Glycolysis being broken down into pyruvate is the fastest way, but we also can use nucleotides from nucleic acids, we can use proteins, we can use lipids. So no matter what we're eating, we can use it to funnel the creation of the of ATP, so long as we have those micronutrients that we were talking about, those vitamins that we need. Okay, so how many ATP molecules do we get out of NADH? Well, when we lose all the hydrogens and come back through, we can drive the process of about 3 ATP molecules per. So actually I wasn't giving you anywhere near the correct numbers when I was saying this was changed and this was the 20. It was just more that this is the one that we use more often, although the NADH can be broken up into about 3 of the ATPs. I'm just saying that the $20 bill is the most common currency. So overall, from each molecule of glucose, we're going to be making 34 ATP molecules. Remember, previously we were only making 2 from aerobic respiration. So that's going to be, I don't know what, 15-fold the amount of energy that comes by being able to take that pyruvate molecule, sending it through the Krebs cycle, and utilizing the ability of mitochondria to perform aerobic respiration. All right, so this is just some basic math here. How many ATP molecules we make from a fatty acid that had 18 carbons? And that's say the 114, and that's because we were making 34 from 6, so we were just multiplying that out by 3. All right. So anaerobic respiration is, again, when we do not have oxygen available. And in this case, we can only use glycolysis to make ATP. And in this case, we're only going to be making, again, about 2 ATP per sugar. And we've got this pyruvate that we don't know what to do with. So the pyruvate is actually going to get entered into a fermentation process. And the hydrogen atoms that are created by glycolysis are going to be donated to organic molecules so NADH can be recycled. So glycolysis can be continued, and we're going to allow, by fermentation, the ability to, again, recycle that NADH so that we can continue this cycle. And there's multiple different types of bacteria that are able to do this. Yeast is also able to do this. Bacteria can perform over a dozen different types of fermentation. Eukaryotic cells, that's us guys, multicellular organisms with nuclei. They're capable of only a few different types of fermentation. So certain things like yeast, which are single-celled fungi, we use quite often for fermentation in order to produce something that we humans love called ethanol. We do that by the conversion of pyruvate into acetaldehyde, and then when acetaldehyde accepts hydrogen from NADH, then it's going to produce NAD again, which again we're going to want to utilize in that recurring cycle. Since we don't have oxygen, we're going to need to use this over and over, and that's going to produce ethanol. However, in animals, that's us guys, pyruvate is going to accept the hydrogen ion from NADH, producing NADH and lactate. So instead of making ethanol, we're making lactic acid. And anyone who's ever had a Charlie horse knows what I'm talking about. So basically what happens when the lactic acid builds up in your muscles, it's going to create a good bit of pain. And that happens generally when you're overexerting the muscles and you've gotten to the point where you need more energy than you're able to use through aerobic respiration, and so you start utilizing anaerobic respiration, which again leads to lactic acid buildup, and we call that a cramp or a Charlie horse. All right, so here's what happens during anaerobic respiration. So here's the process of glycolysis. During the process of glycolysis, we're going to make pyruvates, right? If we're making pyruvate and the pyruvate is not going into the Krebs cycle, then it's going to end up being converted into lactate. In that way, this NADH that's produced from glycolysis can go back to NAD over and over and over so that we can utilize this again to make more ATP. It's going to, again, cause us to create lactic acid, which is a toxic buildup in our muscles. And here's what I was talking about, the alcohol fermentation in yeast. Again, we're making those 2 pyruvate molecules. Those pyruvate molecules can then get converted into acetaldehyde, creating 2 carbon dioxide molecules, which is part of fermentation, right? Carbon dioxide bubbles. And what else is part of fermentation? Acetaldehyde being converted into ethanol, right? So this is how we do our malt liquors, our beers, etc., via alcohol fermentation in yeast. All right, so fermentation in yeast and bacteria is used to produce all sorts of stuff that we love. As I mentioned, wine, beers, but also breads and teas, yogurts, sauerkrauts, kefir, kombucha. So lots of things that you might not necessarily think of because we focus mainly on the ethanol, but it's going to produce the bubbles in the bread, and in cheese is going to be able to, in Swiss cheese, for example, the bubbles. So fermentation is going to be really important for anaerobic respiration. All right, so how do we regulate metabolism? We regulate metabolism by taking a look at what we have and making sure that we have enough. So if we have too much of something, we're going to store it aside as something else. And if we have too little of it, we're going to create more of it. And so if, for example, we don't have enough ATP, our muscle and our liver cells will break down fat in order to create ATP. That's called beta oxidation. We can also, in our liver, convert fatty acids into glucose. That's called gluconeogenesis, neo being new, genesis creation of new glucose, right? In order to maintain blood sugar during exercise. So if we're exercising and haven't eaten properly, our blood sugar would normally drop. However, we're able to take fatty acids and make sugar that we put back into our blood supply so that we can continue the process of creation of ATP while we exercise. However, the longer duration or higher intensity exercise is going to stimulate the fat breakdown more quickly, and it's going to, because in anticipation of this, that exercise maintaining, more total calories will be burned off. So you reach a peak period after about, I don't know, 45 minutes or so, where you're really into the serious calorie burning zone. All right, so this is a level, this is a list of hormones that are responsible for metabolism regulation. So lots of different hormones can stimulate the breakdown of fat to use to be able to create ATP or for the conversion to glucose, right? So we use glucose to maintain blood sugar, but also we use it to create ATP. So it's all kind of linked together. But that's going to include hormones like glucagon, epinephrine and norepinephrine, growth hormone, DHEA, and thyroid hormones. So as you can see, there's a lot of different hormones that regulate metabolism. And the one that we've talked about extensively, and we'll probably talk about again, is insulin. So insulin is going to be the protein that's going to cause cells to convert glucose into fat for storage, and it happens after we eat. Why? When we eat, we're going to eat sugar, and that sugar gets into our blood. When our blood sugar increases, insulin detects that our blood sugar is too high and is responsible for taking the blood sugar out of the blood and storing it in the cells for energy usage, as well as converting it into fat for storage for later. So again, that occurs generally after eating. And it is also what goes wrong in individuals who are experiencing diabetes. Okay. So this here is the application answer. Again, it's pretty long, so I'm just going to let this screen sit here for a minute or two, and then we'll end the lecture. Well, thank you very much, as always, for coming in class and listening to me speak. I will see you again next time, and as always, happy studying. Have a great day.